but we're gonna get started here. I was here last time for the review that Jack led, but I'm leading today. My name is Rachel, and I've got a couple things that we're gonna do. First, I have a Kahoot for us to play. So if you wanna join on in there. There we go. And then we'll get started in a second once we got people joined. If you guys want to, can you grab these packets from here? Just waiting for a couple more. We're almost there. All right. Everybody in? Good. All right, we'll get started. All right, which of the following is not a feature of a bacterial cell? All right, correct answer is nucleus. All the rest of them are found in a bacterial cell. All righty, let's go. Cell membranes consist mainly of what? What are they physically made out of? Awesome. Lipid bilayers. Can anybody tell me? So they have the two parts to them that are come together. There's the head and the tail. How do we describe them? Sure, the phospholipid bilayer, but there's a what kind of head and a what kind of tail? Which one's which?
Hydrophilic is the hydrophobic is the tail.
Yeah.
Protein is the head and the lipid is the tail.
Sure. So we were looking for the hydrophobic and hydrophilic. So hydrophilic is water loving. Those are on the outside and the hydrophobics are on the insides. So that when they two come together, the hydrophobic go on the inside. All right. All right, let's go. What kind of cells are sperm and egg? Good. Yes, they are haploid cells. Someone tell me what the difference between a haploid and a diploid cell is.
Half the chromosomes.
What do you mean by that?
The haploid has, the diploid has the full set of chromosomes.
Sure. So you look at the chromosomes, they look like that X kind of shape. That's the diploid. When it's just the one strand, that's the haploid cell. That's how to differentiate between those. All righty. Yeah, you guys are crushing it. All right, which of the following is found only in a plant cell? Nice. Chloroplast. So that's gonna be where photosynthesis essentially takes place and all that light energy gets converted into glucose or sugar that can be used throughout the plant. Where are proteins modified, sorted, and packaged? Think about this one. All right, good. So that's different from the ribosomes where the proteins are made. The Golgi is where they're going to be packaged. Where is the energy from carbohydrates transformed into ATP? ATP is energy, if that helps you at all.
That's what's wrong.
That's okay. So yep, that's your mitochondria. So you've heard it 1,000 times, but powerhouse of the cell, where all the energy is made right there. Cell organelle that acts as the janitor by recycling cellular waste. Good. So the lysosome is the janitor that does the recycling. The vacuole, that's the one that's just storage, stores the different materials. Nice work. All right, when a plant cell is placed in a hypertonic solution, which of the following will occur? Hyper. So the cell will shrink and shrivel up. Can somebody tell me why that will happen?
Water.
Why would that happen?
I'm trying to work this right. Is it basically because it's gaining more, it's having less solute inside the cell? No, I'm not.
You are on the right track. So it's all about concentration. So the concentration inside the cell is going to be less than outside. So the water moves from less concentrated to more concentrated. So the water is going to leave the cell, making it shrivel up. All right, next one. Ooh, nice. You got people moving up. What do all cells have? Plasma membrane. So remember the nucleus and mitochondria, those will not be in prokaryotic cells. That's the difference for those. And then none of you said it, but why is cell wall not the right answer? Right. They're not, they're just plant cells. What is the process by which a cell uses its plasma membrane to engulf a large particle? Good. That's the phagocytosis. So pH makes like an F sound. I correlate with food. Same sound. So it's like cell eating. The large particles like food. Alrighty. True or false? Sister chromatids are genetically identical to each other. That is true. So what is it? Meiosis or mitosis, where you're going to have those identical sister chromatids at the very end? Good. Mitosis, that's the identical. Right, the period of DNA replication in the cell cycle is called what? That is your S stage. So the G1 comes first and that's just the growth. And then it moves on to the replication and follows through with the G2 and then moves on to the mitosis part of it. What two processes are involved in mitotic cell division? Some of them are similar, read carefully. Nice. So nuclear duplication and cytoplasmic division. The reason it's not the duplication of the cytoplasm because there's no more being made. What's there is just being separated. So you guys got that one. During meiosis, what happens to the number of chromosomes? It divides in half. We'll get to that one a little more later once we talk about mitosis versus meiosis, but whatever number of chromosomes you start with in meiosis, it ends up in half. What is the main difference between mitosis and meiosis? Good. Mitosis makes genetically identical copies. Meiosis does not. That's kind of what we talked about a little bit before with the genetically identical sister chromatids. Nice job. True or false? A zygote always has the haploid number of chromosomes. Good. False. Zygote has the diploid number always. Nice job. In what phase do two nuclei begin to be seen and a cleavage begins? Perfect. Good. Telophase. And then that would potentially move on to the cytokinesis from there for the complete separation. What process does this show? There will be a picture. Oh, I'm sorry, that is very small. The numbers should clue you in though. All right. Good. So that is meiosis. So there were 4 daughter cells there compared to the 2 cells that would be produced through mitosis. Nice. Getting to the end here. Crossing over happens at what phase of meiosis? Crossing over. Prophase. So can somebody tell me what crossing over is? In the back? Yeah, so exactly right. Those chromosomes are gonna come together and they can mix. So I don't know if in your notes, there's the single haploid cell and it has like a different colored top from the bottom. That's crossing over when the pieces kind of exchange a little bit. All right, good. And then last question for the Kahoot. Homologous chromosomes separate during what? That's going to be during meiosis one, that very initial. All right, let's see who our winners are here. Claudia, nice work. Is it Maria? One of them. Megan, good work. Unfortunately, I do not have a prize for you, but you have the pride knowing that you won Kahoot. Congratulations. Yeah, I can send it to you afterwards. Absolutely. Oops. Second. Okay, let's get back here. All right, perfect. When you walked in, there's a packet over there. If you didn't grab it, I can grab you one. But that first sheet, you can work on it by yourself with the people around you. This is similar to that sheet that you have in your notes with the cell as a factory, but it's different. So just number 1 through 10, what you believe the parts of the cell are that are being described with these. I'm going to give you about 5 minutes to do that. If you get done before, we can get started, but I'll give you some time to work on that.
Just the front page or just?
Yep, just the one that looks, says manufacturing factory at the top. Just do that one first. If you can, try not to use your notes. That'll help you study better. Give about 1 more minute here. All right, finish up your thoughts and ideas, write them down. We're gonna go through these. So someone tell me what they think number 1 is. That's the one that says the fence around the factory complex. Go ahead.
So,
What did you say?
so,
You said cell wall. That's your guess.
it was so what was remembering, it was confusing.
Sure. So on here, we've got, hello. Cell membrane. Depending on what type of cell it is,
Yeah, that's why I was like.
cell wall could be an option there, but we're gonna go with cell membrane. This is just gonna be general for eukaryotic cells. All right, number 2, the warehouse. Can somebody else tell me what that one is? Anybody have an idea? It's okay if you're wrong. Yeah,
Is it vacuole?
it is the vacuole. Why'd you say that?
It is, I'm right.
You're correct, yes.
Vacuole store like water and stuff like that.
Yeah, so that's basically what they're for, is just for storage in the cell. Awesome. Number 3, the powerhouse. Nice, nailed it, you guys. So I know we always know it as the powerhouse, but what's really going on is that energy is being converted into ATP, which is energy. That's why it's the powerhouse, makes all the energy. All right, number 4, the packing plant. Golgi body. Why do you say that?
What I basically remember is I'd like to make a little fun little thing about stacking pancakes and other like little packages being stored out.
All right,
It's
so
not my fun little one.
the Golgi body or Golgi apparatus, it preps the proteins and lipids for use in other places. So yeah, it's like packages them up so they can be shipped off to use at other places. Somebody else, number 5, this could be 2 things, so I'll accept either one. Any ideas? The manufacturing plant? Anybody? Ribosome, cool. That is what I had initially. There could be a different option. If it was a plant cell, then it could potentially also be chloroplast. There wasn't really a key going with this one, but it could be either one. So the ribosome is going to be where those proteins are made, and then chloroplast, like we talked about before, is where light energy is going to be converted to sugar. So number 6, that is our foreman. What are we thinking that one is? This one might be a little tricky.
The nucleolus.
Awesome, nucleolus. So do you know what happened, what the nucleolus does?
Yeah, it's the small openings.
Bacterial.
Something is made in the nucleolus. That's very important.
Oh,
Okay,
RNA.
protein force.
Mm, yeah,
Messenger, is it the messenger RNA?
it's the rRNA, ribosomal RNA. It also makes ribosomes. So that's the big thing to remember with the nucleolus. We'll talk about it in a few more, but there's something I want to come back to with that one. Number 7, the data storage room.
Oh, where is it?
Number
So it's
Like
on the
DNA.
That's specifically correct. I need a little more general.
Oh,
Where do you find DNA?
And so,
nucleus? No, wait, it manufactures DNA.
Anybody? It's not necessarily an organelle. All All right, you guys are close with DNA. I just generally had chromosomes,
Oh,
but that is where the DNA is tightly packed within the nucleus. So it kind of,
Because when I think of chromosomes, I just think the short string DNA and mitosis.
so the DNA is in chromosomes. That's what we're thinking here. Okay, so then number 8, the executive office.
Nucleus.
Perfect, nucleus. So what I was gonna come back to with the nucleolus, that one can be hard to distinguish. Just remember that it is within the nucleus.
mhm.
That's the big thing to remember. Number 9, some people looking at it coming in were confused about this one. If you don't have it, it's okay. Did anybody have an idea?
Nuclear envelope?
Nuclear envelope. So you guys agreeing with that? Cool. That is also what I had. Dr. Seleska wanted me to emphasize that it is not a wall like this says, it is a membrane. Things can pass through. It is a membrane, not a wall. All right. So then the last one on here, number 10, the roadways within the factory. What are we thinking that one is?
I can't say it, but I just want to use
ER is fine. Yep, endoplasmic reticulum. So somebody tells me, I guess this kind of says it, but what does endoplasmic reticulum do in the cell?
Or it's basically guide proteins. Because I know what it does, it just.
Yep, so it transports compounds throughout the cell. Good. All right, nice. In that packet I gave you, what's the sheet that goes after that?
Comparison of
Comparison.
bacteria.
Okay. So for that one, I'm gonna give you some time to work on that one. I'm gonna give you kind of a short amount of time, but we'll go through it together. So just kind of in these boxes, you're gonna put yes or no if those things are present in there. So yeah, you can work with people. Take some time to do that, and then we'll go through there as well.
Is this the beginning of the document?
Mhm.
Oh, sorry.
Yeah, what's on there right now has nothing to do with what you're doing, so you can ignore it. I'm just gonna give you about another minute here to get some initial thoughts down, but we'll go through it together. All right, let's go through some of these. So we're gonna start with the prokaryotic cell column. Can somebody just tell me what a prokaryotic cell is? Like, how do you distinguish it from a eukaryotic cell? There's kind of like two big things.
Doesn't have a nucleus.
Great, that's the one I was looking for. Do you have the other one?
Doesn't have a cell number.
That is kind of true. So the two things I'm looking for is it doesn't have that nucleus. So for that one, you can put no. And it doesn't have any of the membrane bound organelles. So from the nucleus down, everything in that column is no, except for ribosomes. There are ribosomes in prokaryotic cells. They are just small.
Isn't it nucleolus?
I think it's nucleoid for that one.
Right, there's a nucleoid.
Because nucleoid is the bacterial chromosome that's located inside the bacteria.
Yeah,
What do you, instead of what?
For prokaryotic.
that's there.
It doesn't have the membrane bound nucleus. Yeah.
Okay.
Yep, that's what it is. So for cell wall, it's not always, but they usually have it. I don't know if this is important to note, but Dr. Sluska had on his notes that the cell wall in prokaryotic cells are made out of peptidoglycan. And yes, they do have a plasma membrane. Okay. Yeah, sorry, question.
Prokaryotic cells actually have cilia and flagella. No. Wait,
Sorry, you were correct. Yes, they do. Yep. Good catch.
I just want to make sure.
Yes. Nope, you're correct. I missed that. That one was at the end. Okay, so then eukaryotic cells, in the animal column, can somebody tell me what they don't have? Because they have most of these things. What do they not have? Chloroplast and a cell wall. What about the cilia or flagella? Do they have that?
Yeah.
I thought it was sperm cells.
So they oftentimes have it, but not 100% of animal cells have them. So it's most of the time, so you could put like often. They usually do, but it's not that all of them have to have that.
Like the respiratory tract has the cilia.
Yep, good. That's a very good example. All right, the plant column. So we said the animal cell does not have the cell wall, but the plant cell does. Can somebody tell me what a plant cell wall is made out of? Great, cellulose, perfect. What else on here? What do plant cells not have? Because again, they have most of these. Lysosomes, good. Plant cells do not have those.
Wait, live flagellas? I don't think they do.
So that one, he's not gonna ask you on that one. That one depends on the plant cell. So like flowering plants don't have them, but like bryophytes and ferns and that kind of stuff has them, but I don't think you need to know that in depth about it. There's one more that they do not have.
They got mitochondria.
They have mitochondria.
Cytoskeleton.
No, they've got that.
Centrales.
Centrioles, yep. That is one they do not have, is the centriole. Alrighty. Cool. All right, so we're gonna take a break from that packet for just a second, and I got a PowerPoint for us. We'll go through a little bit. The cell theory, this is just important to know. All living things are made of what? Cells, great, that's an easy one to remember. Why does this never want to work? Cells are the basic what? Unit of life. So all living things are made up of them. All cells come from where or what? Great, pre-existing cells. This one might be a little weirder unless you've been up with it, but what occurred within cells? No. I'll give you this one because it's gonna be weird to come, energy flow. So that's gonna occur within the cells. What is passed on from one cell to another? It's a certain kind of information, traits, DNA, sure, kind of both hereditary information, but those are all kind of what you said clumped into the same idea. And then all cells have the same what? What do they all have in common? No, prokaryotic cells don't have that. This one I can give you. It's just the basic cell composition. So they all have a similar kind of structure that they have going into it. Alrighty. Do you need this up for another second? Alrighty. So I've got listed here on that side, the different proteins that are in the plasma membrane. They get matched to a description on the other side. So I need one person, if you can read, what does channel proteins match up to on the other side? Yeah, in the back. Great. Why is it not working? Yes, the channel proteins are allowing ions or molecules to freely pass through the plasma membrane. So all these proteins, remember, are within that plasma membrane somewhere, so they're functioning through it. All right, someone else, what is the function of a carrier protein? Yes.
It allows water cells to go in. No, it's not on the water cells, solubility.
Say what you were going to say before, and I can maybe refine what you're going to say. Can you find one that fits something I have up here? Or if anyone else has an idea.
No, wait, recognizes and wanted cells. Wait, no, that's recognition. I know what it is.
What were you going to say about water before?
It's soluble in water, allows soluble water cells to pass through it. It's just a
Okay,
process.
okay. So you said water cells. The key thing in here is specific ions. So water is a specific ion that is allowed to pass through the carrier proteins. And there are other carrier proteins that are specifically for sodium or specific for potassium.
They use ATP.
Correct. We'll get to that later, but you are correct. So yes, a specific ion so it can cross the membrane. So the channel proteins just kind of allow everything to come through, but the carrier proteins are more specific. All right, someone else, can you tell me what matches up with the cell recognition proteins? This one is easier because great. Yep, so it recognizes only wanted cells and attacks those without the proper glycoprotein. So I don't know if you've seen pictures of those ones, but they're usually depicted as having a little, like it almost looks like an antenna kind of sticking out. And it's looking for ones that have that same antenna kind of looking structure. And if it recognizes a cell that does not have that, it realizes that it needs to attack that because it could be a harmful substance to the body or whatever. And so that's how your body knows to take care of that in such a way.
So that's where autoimmune disease comes.
Correct, if those aren't functioning properly. All right, someone else, can you tell me what a receptor protein matches up to? Awesome. So those are ones, you know, if the top of it has a circle opening, it has to match with that like a key in order for it to come through. Or if it's like a square kind of shape, they're not really going to be that shaped, but that's kind of, it can only receive a certain shaped molecule. And then our one left are enzymatic proteins. So that'll carry out a specific reaction and catalyze it. Do you guys know what it means to catalyze the reaction?
Mhm.
What does it mean? Speed it up. Great. Sure.
Sorry, my anxiety's overflowing.
You're good. We already did the, I'll go back to it in a second. Just give me a second to see what we got next. Okay, we already did that. Perfect. Yep. So I'll leave this up so you can go through it a little bit more. Then the table you have that says effect of osmosis on a cell should be in your packet. You can take a picture, finish writing this one, and then I want you to fill out that one that you've got. Give you a minute or two to do that one. It says like tonicity of a solution, that kind of thing. Are you guys all good with what's on the board? Can I take that down now? Okay, cool. So for the first couple columns, I'm just looking for things like it's more than the cell, less than the cell, same as, which are kind of things I'm looking for. Alrighty, let's go through this one. So in an isotonic solution, the concentration of the solute is what compared to the cell? Does it not like to work? Correct, the same as. What about hypotonic?
Lower, wait, higher, higher.
First instinct.
What? No.
Lower.
Because hypotonic.
Okay, we'll come back. So then hypertonic must be more than. All right, so that's the concentration of the solute. So then the concentration of water in an isotonic is also the same. Yep, isotonic's really always gonna be the same. Hypotonic is more in this case. And then hypertonic less. So hypo in prefix world always means less. So you think of like hypothermia, your body temperature is less than what it should be. Hypertension is if you, yeah. Yep, good connecting words. But it's less than when referring to the solute. If you're referring to water, then it's the opposite. All right, so the net movement of water, so I'm just kind of asking, does the water move in or out of the cell? Isotonic, what about that one? Right, there's no movement of water. Yep, it's equal. Hypotonic, does the cell gain water or does it lose water? Great, the cell gains water because there is more water outside the cell. Water wants to move from areas of lower concentration to higher concentration. So then the hypertonic cell would lose water. So then what happens to the cell?
Nothing.
Nothing. Hypotonic, does it swell up or does it shrink? Great. It swells up. So in a plant, that would be turgor pressure. So that's why like plants are able to stand up. The stems don't just fall over. That turgor pressure of water swells, keeps it up. And then hypertonic, it shrinks. So that's plasmolysis in like your body cells. Awesome. I'm gonna go back to this one. Talking about passive transport and active transport. So through your plasma membrane, we kind of talked about with the different types of proteins. but molecules need to flow through that membrane. So passive transport does not require energy. It can just flow through by diffusion, doesn't need any help. So that's like, it says no ATP, that's your energy. And then active, same process, but they have to be pumped through by another source. So that energy is required in that case.
So would the example be the sodium potassium pump in the here?
Yes, great example, the sodium potassium pump. That is active transport, correct. Alrighty. So I've got a few slides. I gotta get to them. On meiosis, mitosis, let's get there. All right. So meiosis, the cellular reproduction, which the number of chromosomes are reduced by half. Remember we saw that in the Kahoot. Through the separation of homologous chromosomes, producing haploid cells. That's key for this one. So, meiosis 1, what are the order of phases kind of called?
Oh, wait, what?
So what the phases of meiosis?
Oh, prophase, metaphase, interphase,
Oh.
phase, and then part 2, the other part in the second.
So yeah, so you see here, we've got the prophase, metaphase, anaphase, which is right here, and then telophase. So
Oh,
in this one, it kind of more in depth describes what happens. So in prophase 1, that chromatin is going to condense, and you see in the that nuclear membrane is going to disappear. Metaphase, the homologous chromosomes line up on opposite sides. So you can see those spindle fibers kind of pulling them apart. Anaphase, the chromosome pairs separate and move to opposite poles. So they're kind of separated into their 4 quadrants there.
I'm too embarrassed to ask you to go back because I don't want to waste anyone's time.
We can go back to it after and I can get you caught up